Good day to everybody. Thank you for joining our webinar today. I'm going to be presenting on a novel technology called switchSENSE that employs electro-switchable biosurfaces for the biophysical analysis of biomolecules. We're from the company called dynamicBIOSENSORS, which is a technology provider. Our R&D headquarters are located in Munich, and the main offices for our US subsidiary are located in San Diego, California. And as you can see on the map, we also have global sales representation in many different countries and different cities. Our unique switchSENSE measurement technology can be used for the measurement of many biophysical parameters, as I'm sure you already recognize quite a few from the slide that you have in front of you at the moment. Some of these overlap with other biosensor technologies, for instance, including, kinetics parameters such as K on, K off, and KD. But also we are able to probe other unique measurements and unique parameters. And these include dual target immobilization, thermal stability measurements, and the determination of target size and conformational changes. The basic switchSENSE principle is that we rely on DNA-based biosurfaces. So we tether DNA to gold electrodes, and one of our DNA strands carries a fluorophore. That fluorophore serves as a reporter dye, and we track the fluorescence signal from this fluorophore in order to run our measurements and determine all of these biophysical and kinetic parameters that I had just mentioned to you earlier on and rather briefly. We also rely on time-correlated single photon counting in order to track how the fluorescence signal changes as a function of time. This is what our switchSENSE analyzer looks like. So in the center over here, you're going to see our device, and this is what we refer to as the DRX. We have both a DRX and a DRX². Within our switchSENSE analyzer, we have an autosampler, which has a lot of temperature control features, so you can control your sample storage temperature. We have integrated microfluidics and liquid handling systems, where we're able to control flow rates from very low rates of down to 5 microliters per minute up to very high flow rates that reach up to 4,000 microliters per minute. The device is also fully automated, so you can program whatever experimental script you would like to run, and the device runs automated for up to 72 hours. The sample measurement itself takes place on a biochip, and we also have temperature control features for the biochip itself, and those are independent from the temperature control of the autosampler. So you can run the experiment at a different temperature from that which you also store your sample. I won't go into the details of all the instrument specifications, but I invite you to visit our website if you're interested to learn any further details about our instrument specifications. The interface actually is very friendly and rather intuitive. On the left-hand side, you'll see that in order to run an experiment on the DRX analyzer, what you have to do is load your buffers to the left hand of the device, load in your vials that contain all of your samples, as well as the biochip, and you're ready to go in order to run an experiment on the DRX. Now, the versatility of the DRX and our ability to actually run so many different measurements and probe very many different biophysical parameters is awarded by the fact that we're working with DNA-based biosurfaces. DNA is a very versatile molecule. We can very controllably tune what we functionalize onto the DNA, and we can engineer and create very many different biosurfaces that allow us to study very many different systems. So on the top, you'll see schematics that represent very many different ways in which we can immobilize target biomolecules on the surface before running, again, the biophysical analysis, as I had mentioned, with a list of parameters that you see in front of you that include kinetics parameters, IC50s for small molecules, protein diameters, conformational changes, as well as melting temperatures, TM for thermal stability. So now that I mentioned that we are a biosensor-based technology, the first thing that we have to do in order to run our experiment, in fact, is to immobilize our targets onto a chip. So let's start with a little schematic of what our biochip looks like. In this case, we have 4 independent flow channels, so each has an inlet and an outlet. They're independent, so there is no crosstalk from one to the other. And each of these channels houses 6 measurement electrodes. And so we're going to see how we can actually immobilize our biomolecules, our targets, onto these electrodes in order to run the measurements. So as I mentioned, what we have gold surfaces. Those are the measurement electrodes themselves, and we refer to these as electrodes because we can apply voltages at the gold surface. The gold surfaces will carry single strands of DNA that are functionalized with a fluorophore, as we had introduced before. That is our signal source. That is our reporter dye that we're looking for. Now, we take advantage of the highly efficient process of DNA hybridization in order to couple our ligands or our biomolecules of interest onto the surface. And the way that we do that is that we want to couple the ligands themselves to the complementary strand of DNA, here represented in blue. Once we couple them together, we hybridize that onto the surface, and by that you've created, or you have moved your ligand and immobilized it onto our DNA-based surface. Now, we can do this in one of 2 ways. We can either covalently couple the ligand to the single strand of DNA, and the way to do that is by using standard chemistries, including amine chemistry and thiol chemistry, for instance. And then, so the same way that you would add, for instance, a fluorophore, or that you would conjugate any protein to a specific fluorophore, you can also do the same thing with the DNA. And the way that this step looks like, it's also a very straightforward step, and we have coupling kits that also help our customers go through the entire step of the DNA protein conjugation. The first thing involves activating the DNA with a cross-linker, that it is reactive to your target. The second thing is that you add your target to the activated DNA, incubate the reaction, allow it to proceed, and then purify the unconjugated DNA, so DNA that has not coupled to a ligand, from DNA that is in fact coupled to a ligand. And we do have our own protein purifier series, the Profire as well. That is a tailored system specifically for the purification of protein DNA conjugates. After you're done with the purification of your conjugates, you're able to create as many stock solutions of that as you want, place it in the -80, and essentially take out 1 stock for every experimental run that you will do. Now, you don't always need to covalently modify your protein in order to immobilize it onto the biochip. You can also take advantage of capture methods. So, for instance, very many different proteins are expressed with his tags or that come biotinylated. What we can do is we can also create capture DNA, such as NTA DNA, hybridize that on the surface, and then use that for the capture of your his tagged protein. Now, the advantage of all of this is that we can actually use a wide range of affinity-based immobilization methods. It enables you to use an unmodified version of your protein. And again, you can immobilize your target in a manner which enables it to be most stable and most appropriate for surface immobilization for the specific assay that you're interested in running. Now, what the device does after you've created the conjugate, so the creation of all of these protein DNA conjugates, that is a step that comes offline. So after you've created those conjugates, now what does the DRX do? So how do I move from putting my ligand on complementary DNA? Now, how do I put it on the biochip itself? And this is where the DRX comes in. And here I'd like to introduce to you just a standard measurement experimental cycle that the DRX conducts. In the first step, in the first diagram that you see over here, you have a single strand of DNA that is carrying a red fluorophore. The DRX itself will be programmed in order to pick up your conjugate sample and then hybridize it onto the surface. By that, it automatically functionalizes your biochip surface with your ligand of interest. As a second step, the DRX will also be programmed in order to aspirate whatever sample you have. In this case, I'm representing the analyte as an orange antibody. It will flow your analyte over the surface and enabling you to study the association kinetics of your antibody, in this case to the ligand. After that, you can switch from analyte into buffer solution and then proceed and study the dissociation kinetics. And one very unique thing about our biochips is that you're always able to regenerate them. They're not a one-time only use biochip. What you can do is that you can dehybridize the DNA and then regenerate the chip to its original state. This means that you don't have to worry about the stability of your ligands on the surface because you can simply strip the surface and start anew, and you're not dependent on the stability of the ligand that you had for long-term measurements. We have, as I mentioned a little bit earlier on, we have both a DRX and a DRX2. And the difference is that the DRX has one LED, a red LED, and one detector. But our DRX2, for the dual color detection, we have two LEDs, both a green and a red, and we have two detectors, again, for the simultaneous detection of two antigens on one detection spot. And here is just a schematic representation again of our biochips and our measurement electrodes, and then a zoom in as to what those electrodes look like. Each of those red dots represents a DNA nanolever in the case of the single color chip, and the green and the red dots represent both green and red labeled nanolevers, in this case for the DRX2 or the dual color chips. Please note that the sequences of the DNA for both the green and the red nanolever are different, which is why you're also able to conjugate or functionalize two different entities onto the surface for a dual detection measurement. Now that I've given you a quick introduction as to how you can immobilize your targets on the surface, now I'd like to describe to you what our different measurement modes look like. So I mentioned to you that they were electro-switchable biosurfaces, so we have DNA that is coupled to a gold surface. We can apply a voltage on that surface, but what is it that we do with this DNA that enables us actually to run the biophysical analysis for all of these intermolecular interactions? To start off with, I'd like to introduce to you our first measurement mode, which is referred to as the static measurement mode. In this case, what we do is we apply a negative voltage onto our gold surface. The negative voltage repels the DNA, and as a result, the fluorophore remains far from the gold surface. For this, what we do during this measurement mode is we simply measure the change in the proximity of this fluorophore to give us indication about binding kinetics and binding parameters. So the way that this works is that the fluorescence or the quantum yield of the dye is going to vary based on the presence or the absence of an analyte in the proximity of the fluorophore. Just to give you an example to illustrate here with a little movie what this would typically look like in terms of a measurement and what the readout would actually be. So here we have our flow channel, we have our DNA nanolevers carrying the fluorescent dye that are functionalized onto our gold surface, and here we have a ligand represented in red. When we apply a negative potential, the DNA nanolevers are held away from the surface. When we inject our analyte, the analyte binds to the ligand, and the proximity of the analyte binding to the ligand actually quenches the fluorescence. And we detect this rate of quenching as the association kinetics. When we switch back into buffer and the analyte dissociates, we have a recovery in the fluorescence signal, and this fluorescence or rate of fluorescence recovery represents the dissociation kinetics. We simulate this K on and K off from the plots of the association and the dissociation, and we use that in order to determine the KD of the interaction. Now in the second measurement mode, we don't only apply a negative potential on the surface, but in fact we apply alternating potentials, and we refer to this as the dynamic measurement mode. Now, the proximity of the fluorophore to the gold surface quenches it. So we're actually able to track the location of the nanolever relative to the surface by using the quantum yield or the fluorescence signal itself. And again, just to illustrate what would this look like in terms of an experiment and how do we use this readout in order to give us information about kinetics of binding. Again, the standardized flow channel over here with the gold electrode, our nanolevers also carrying both the fluorophore and the ligand. In this case, we're applying alternating positive and negative potentials. The positive potential attracts the DNA nanolevers to the surface and the negative potential repels them away. The introduction of analyte into the solution is going to bind onto the nanolevers and slow down their motion. So essentially what we're doing is we're looking at how fast these nanolevers move, and this gives us information about the cargo that they're carrying. So when the analyte dissociates, the nanolevers are now lighter. They carry a smaller load, and as a result, their switching speed increases again. So again, we simulate the drop in switching speed as the K on, and then the increase in switching speed as the K off in order to determine the KD, or the kinetics of binding, for this interaction. Now, the interesting thing about the dynamic measurement mode is that it's something which is rather unique to the switchSENSE technology, not only in the fact that we can move or actuate the DNA nanolevers that we have on the surface, but also because we can use the motion and the rate of motion in order to give us information about the shape and the size of targets that we have immobilized on the surface. As I mentioned earlier, in the absence of any target which is bound to the DNA nanolevers, the DNA nanolevers themselves are going to have very little viscous drag in the solution, which means that their motion is very rapid. So fluorescence recovery as a function of time is very fast. But whenever we add a protein, or whenever we add a ligand, what we're doing is that we're slowing down the motion of the nanolever. So what we do is when we want to run a sizing measurement, we compare the motion of the nanolever in the absence of the ligand to the motion of the nanolever in the presence of a ligand, in this case represented as an orange protein, and we compare this motion to a library of solutions of theoretical curves from simulations that we have generated, and we use that to give us information about the hydrodynamic diameter of the protein which is attached onto the DNA. So with that, I would like to give you a couple of examples of all the different applications that we have. So there are quite a few, but I will only focus on some of them, and I will show you some exemplary data from each and every one of these applications. So to start off with, I'm going to discuss the analysis of binding kinetics using switchSENSE. Now, since we're a DNA-based biosensor, I guess it won't come as a surprise to everybody that one of the things that we do study is small molecule ligand binding to DNA. And the first example that I would like to use in order to highlight that is the binding-dependent folding of a cocaine DNA aptamer. So in this case, what we do is that we hybridize the aptamer onto the surface. We associate a small molecule ligand. We study the folding of the aptamer as a result of binding of the small molecule ligand, and then study the dissociation, or then the loss of structure of this aptamer again. In this case, again, we used a small molecule ligand. We flowed that over the surface as the analyte at increasing concentrations, so from approximately 5 nanomolar to approximately 20 micromolar. And what we studied in this case is the change in the fluorescence signal. So in this case, the folding of the aptamer, whether it was unfolded or folded, changed the proximity of the fluorophore, and we detected that as a quenching in the fluorescence signal itself. And so what we did over here is we did this as a function of concentration in order to run a global fit of the series for an accurate determination of the K on. And then we also used the end levels of the association and fit that using a 1-1 Langmuir model. Again, these are capabilities that are enabled by our analysis software. And from that, we determined the KD to be approximately 600 nanomolar. And this is something which matched very well to the literature reported ITC value. Now, another example, really a broad set of applications for us include DNA and RNA binding proteins. And I'm sure many of you have heard of the CRISPR-Cas9 system. Here I'd like to use an example of a study where we tried to probe the fidelity of recognition of the Cas9 with the mismatch position within the guide RNA. And the way that we did that is that we were given various different guide RNAs, and they had mismatches at different locations within the guide RNA relative to the PAM sequence. And the way that we conducted this measurement was that we used our dual colored chips with DNA and RNA binding proteins. It's always of interest to see if you have any unspecific binding, and that is why we ran it using the dual color, because within the same assay, within the same flow channel, you can determine whether or not you have any unspecific binding by looking at random sequences where you do not expect your DNA or your RNA binding protein to bind. And then on the reds nanolever, what we had was we had our target sequences that included all of the different spacers. Now, the convenience of the way that we created our setup is that you can use the same biochip in order to run very many different target sequences on the same surface. So all we had to do was regenerate the target sequence on the red nanolever and then go through all of the different spacers for the iterative cycles of experiments that we wanted to conduct with Cas9. In this case, what we were able to demonstrate with these fidelity tests is that the closer the mismatch was to the PAM sequence, the more affected the binding activity was. More specifically, in the absence of any mismatch, we measured a K-on rate on the order of 1/10 to the 6. But in the presence of mismatches, when it was very far away, that didn't really affect the binding. But the closer it came to the PAM sequence, essentially, we reduced the binding activity, as you can see for the curves on the bottom right, where there is essentially no longer any binding of the Cas9 to the DNA nanolever. Now, not only can we look at sequence specificity, but we can also look at different types of guide sequences. So not only RNA sequences, but also RNA DNA hybrids. And we conducted a project in collaboration with AstraZeneca, which is now published in Nature Communications, where they were interested in seeing if they could create DNA RNA hybrids in the interest of having more stable guide sequences. And what we were able to help them with is essentially show that depending on the hybrid construct that they had created, the Cas9 either maintained fidelity of binding or lost fidelity of binding. So in this case, you can see in the graph on the right is that if you completely replace the RNA with DNA, you essentially knock out any binding of the Cas9 to the recognition sequence. And for general information as well relating to the study, not just of protein DNA, but also protein RNA interactions, there's also a really nice publication that we have generated together with one of our customers, which highlights how the switchSENSE technology can be used for the study of protein RNA interactions. Now, one very unique set of DNA binders also have enzymatic activity, in this case, polymerases to be specific. This is also another feature of the switchSENSE technology, where we're not only able to measure the kinetics of binding of polymerases to DNA, but we're also able to measure the activity of polymerases. And we measure the activity of polymerases in terms of added base pairs per second. And the way that this works is that we're able to track the elongation of a DNA nanolever by the addition of DNTPs and the growth of the DNA by the polymerase, again, using the fluorescence signal, which is tethered to the surface. The advantage, of course, here is that you do not need to use fluorescently labeled DNTPs. So you do not need to use modified DNTPs, as the reporter fluorophore is hybridized onto the biochip surface. So all you have to do is flow in your polymerase, onto a rather unique scaffold that we have, flow in the dNTPs, and then observe the growth of the nanolever or the increase in fluorescence as a result of the nanolever moving away from the surface. This is also something which can be run as a function of temperature, again, thanks to the temperature control capabilities that we have. Again, please refer to our publication in which we as well published our work relating to the polymerase DNA interactions and enzymatic activity using our electro-switchable biosurfaces. Now, we don't just, as I mentioned before, we are a DNA-based biosensor, and we started off with just using DNA as the scaffold or as the binding partner. But as I had introduced before, you can also capture or covalently couple various different proteins onto the surface. And so again, one big interest group for people who are studying binding kinetics is to study protein-protein interactions. And here I'd like to use an experiment or a set of experiments that was conducted to study the binding affinity of interleukin with another protein binding partner. What we did in this case, again, we used our reference DNA. It's always a good idea to have an on-spot reference to confirm that you don't have any unspecific binding. We used streptavidin nanolevers on the red DNA, and we use streptavidin in order to capture the biotinylated protein onto the biochip surface, and this is also something which you can trace as a function of time, so the saturation of your surface is something that you can confirm even before you run your experiments. And then after that, we associated the interleukin as an analyte and studied both the association and the dissociation kinetics. In this case, the binding of interleukin, in fact, increased the fluorescence signal. And of course, the dissociation has to be, the negative signal to the association. And so what you have is then a loss of fluorescence, which represented the dissociation of the interleukin from the protein binding partner. And this is, the standard experiment that can be done. In this case, the interest was to see how various different mutants of interleukin would affect the binding to a specific protein binding partner. And here we ran the assay not only for the wild type, where we had a KD of approximately 500 nanomolar, but also with 3 other mutant constructs. And of course, the interesting thing that you can see here is that using the very same assay, we're able to probe kinetics measurements with KDs from the 3-digit nanomolar to the 3-digit picomolar range. Now, the most common protein-protein interactions are typically the monospecific bivalent antibody binders that a lot of people work with. And again, we have a lot of work on protein or antigen-antibody interactions. And here I would like to show you some exemplary work that we have done with some commercially available antibodies. And again, I would just like to take this data set in order to illustrate how we can probe very fast K-on, so approximately 3×10⁷, but also very slow K-offs and KDs that are even sub-picomolar in the case of embryos. So we can work with very weak binders in the case of micromolar binders, but we can also work with really tight binders due to the stability of a biosensor surfaces for many, many hours. One advantage of working with a fluorescence-based biosensor is that we have very tight ligand density control, which means that we can control the number of DNA nanolevers that we put on the surface. Number of DNA nanolevers that we put on the surface, of course, in this case, would correlate to the density of antigens that we have, for instance, when we're working with antigen-antibody interactions. And the interest in this case is that when you reduce the density of the antigens that you have on the surface, you could effectively, and in the same assay, run both affinity and avidity binding kinetics. So you can play around with the antigen density, and then you could vary the population of antibodies that is bivalently engaged on the surface and a population of antibodies which is monovalently engaged on the surface. Now, this setup where the density can actually be controlled is of high importance and high relevance, for instance, in the study of the affinity-avidity relationship of the FcRn-IgG interaction. So what has complicated really a lot of the kinetic studies of the FcRn-IgG interaction is in fact the density control. And here, this is something that we were able to overcome using our switchSENSE biochips. So in this case, what we did was we created FcRn surfaces. So we functionalized FcRn on the surface itself. And we were able to adjust the surface density, whereby we could confirm that we had approximately a 50-50 ratio of monovalently bound IgGs and those that were bivalently bound, meaning associated to two FcRns on the surface. And we were able to run this study as a function of pH. Again, the binding affinity of the FcRn-IgG interaction is highly pH sensitive, and that is also something which our sensor surface can rather readily do. So we're not sensitive to changes in buffer pHs. This is something that we can rather readily run in terms of an experimental parameter. And here, of course, we ran the affinity analysis first of the FcRn-IgG interaction as a function of pH, and we were actually able to demonstrate that as the pH increased, the affinity between the FcRn-IgG interaction decreased. And this was mainly due to the faster K-off rates that we were observing. So more or less the K-on rates were not very affected, but we were able to measure faster K-off rates as a result of increasing the pH. Now, another thing that we were able to demonstrate, and this is again thanks to the fact that we were able to control the surface ratio, the surface density, and modulate between the affinity and the avidity binding, is that we were also able to show the relationship between the strength of the affinity interaction to the avidity enhancement as a result of bivalent engagement. And here we can really nicely show as well that as a function of pH, so the lower the pH is, the higher the affinity, or the stronger the affinity between FcRn-IgG. And as a result of the higher affinity, we were actually able to measure an increase in the avidity enhancement. So of course here it's very interesting to see that the contribution to the avidity enhancement is also dependent on the strength of the affinity binding mode. All right. Now, monospecific antibodies are also are not the only thing that we can probe on our biosensor surface. I have brought up the dual color chips multiple times before, and one area where this is a lot of interest is actually with bispecific antibodies. And the reason for that is because you are enabled to immobilize two different antigens on the surface, but you're also able to quantify the number of antigens that you do have on the surface. So the amount of antigen that you have there is not dependent on the reactivity, but is dependent on the number of nanolevers that you spot on there. And once it's a 50-50 ratio, then you can be certain that you have 50-50 of red antigen to green antigen on the surface. And this, of course, advances the study of bispecific antibodies tremendously, because you're able to run both affinity and avidity measurements in the same assay orientation. Now, what does that look like experimentally? I'd like to use a movie as well to illustrate to you how the standard bispecific antibody kinetic measurement looks like. In this case, we use our dual color chip with the red DNA nanolever carrying the red dye and the green DNA nanolever carrying the green dye. We start by mobilizing antigen A and studying the binding kinetics of the bispecific analyte to antigen A. And then we immobilize only antigen B and study the kinetics of binding to antigen B. And then we immobilize both of the antigens, and in this case we study the avidity binding, or the bivalent engagement of the antibody to both of these antigens. And so as you can see over here, that using the same assay orientation and using the same two antigen conjugates, you're able to study both the affinity and the avidity kinetics of your bispecific antibody. So, and this is just to show you some data in order to illustrate that. We were provided with a bispecific antibody construct from one of our collaborators, and the desire here was to study the binding kinetics both in the affinity and in the avidity binding mode. So we first start by immobilizing only one target on the surface, in this case target A, the green one, and then here we measure a KD, which is approximately 340 picomolar. And then we immobilize target B, and in this case we measure a KD which is approximately 60 picomolar. So this again was the single target surface functionalization for the affinity analysis. And then we went in and immobilized both targets on the surface in order to measure the avidity binding analysis. And here, as you can see, both in the red and the green signal, we measure the same K off, which is about 0.6, 10 to the negative 4, second minus 1. And the interesting thing over here, and which is a feature unique to bispecific antibodies, but is also a parameter that can be probed using the switchSENSE technology, is that you can also measure the independent K on rates onto the red arm and the green arm. And you can still see over here that despite both of the antigens being on the surface, you're still able to measure two independent K-ons that are reflective of the features of the individual arms of the bispecific antibody. And so here we put together a plot which represents the affinity binding mode, first to the target A, the green target, to the target B, and then as well to the avidity binding. So as you can see that you can measure a large enhancement as well in the avidity binding mode. And here, as I mentioned, that you can get stable kinetics measurements, in this case down to the 2-digit picomolar, so from 60 picomolar down to 17, right? And then approximately a 10-fold enhancement in the binding affinity when you're looking at the green arm in the case of this bispecific antibody. And now, one other thing which has come up and that is of a lot of interest to very many different people is to study target selectivity, for instance, of various different bispecific antibody binders. And one parameter which is of interest to vary is the ratio of surface functionalized antigens. And here the idea is to mimic the expression rate of various different cell surface antigens depending on the various different cell types. So in this case, we're using EGFR and HER2 as an example for some measurements that we had conducted. HER2 is typically the lower expressing surface antigen. So what we wanted to do was we wanted to modulate our biochip surfaces in order to mimic or emulate cells that express much less HER2. And as you can see over here, again, using our dual color, we are able to use this nifty technique where we can use a black hole quencher to turn off some of the red nanolevers while maintaining the green nanolevers. And by virtue of doing that, we're able to modulate the ratio of HER2 to EGFR, or red to green in this case. And effectively, what we expect to do is we expect to effectively change the ratio of monovalently engaged antibodies, so bivalently engaged antibodies and monovalently engaged antibodies, as a function of the surface expressed antigens. So now that I have described, I guess, some highlights for a lot of the different kinetics measurements that we can conduct on our biosensor surface. So again, small molecule ligand binding to DNA, DNA RNA binding proteins, proteins, monospecific and bispecific antibodies. I'd like to bring you to this segment of the webinar where we present actually one of the unique capabilities of the switchSENSE technology, and that is for the analysis of protein size and conformational changes. So again, just a small refresher, as I had mentioned earlier on, that what we're trying to look at in this case is we're going to actuate the nanolever. So we're going to apply positive and negative alternating voltages, and we're going to move the nanolever. And the speed of nanolever motion is going to reflect the friction, the hydrodynamic friction that the nanolever experiences. And we're going to use that measurement of the hydrodynamic friction, or the speed, in order to give us information about the hydrodynamic diameter of the ligand which is bound onto the nanolever. So here We're just going to show you this little movie as well, where you can see that the larger the blue analyte in this case, which is bound to the DNA nanolever, the slower the motion of the nanolever itself. And of course, this scales not linearly, but this scales with the size of the target that we have on the surface. So now we're going to take this information and we're going to convert it into size in nanometers, and we're also going to try to say something about conformational changes. And again, here's one movie in which I would like to illustrate to you how we detect or measure these conformational changes. This is again the schematic representation of our flow channel, of our DNA nanolever in this case, carrying the fluorophore, as well as the target, the ligand in this case, that is supposed to undergo the conformational change upon binding, for example, of a small molecule ligand. So again, we apply an alternating potential in this case, in which we're moving the nanolevers up and down. We flow in the small molecule analyte, and this small molecule analyte in this movie induces a conformational change compaction. It compacts the ligand that we have on the surface. And as a result of that, the hydrodynamic friction that the nanolever experiences is reduced, and we measure this as an increase in the switching speed itself. Now, of course, the very interesting thing over here is that you can measure responsiveness of your target on the surface. So you can immobilize the target in one state, run a sizing measurement, but then you can also measure the real-time change in the conformation upon flowing in a small molecule analyte over your ligand of interest. Now, one example that I'd like to use in order to illustrate that is, in fact, the sting dimer. The sting dimer is known to undergo conformational changes upon binding of small molecules. It goes from an open conformation to a closed conformation. Again, this is something that we were very nicely able to measure using switchSENSE. What we did was we immobilized a sting dimer onto streptavidin, so we used biotinylated sting, and immobilized actually a functional dimer on the surface. And this is something which can be tricky with a lot of biosensor surfaces when you're working with dimers, because it's not always straightforward to immobilize a functional entity on the surface. Nevertheless, here we successfully managed to immobilize the streptavidin the sting dimer onto streptavidin, and we were able to flow in cyclic GAMP, which is a small molecule binder to sting, which binds to sting and then closes the conformation. And as you can see in the left-hand panel, in the open conformation, the string streptavidin complex was sizing around 9 nanometers, and in the presence of the cyclic GAMP, upon binding of the small molecule, this compacted the structure, and we were able to measure a size of approximately 7.5 nanometers. So this was really a nice demonstration. So this is the same target where you measure the same target in an open conformation and a closed conformation. Another example for small molecule induced conformational changes is the transglutaminase 2 target. So transglutaminase 2 is a rather tricky protein to crystallize because it contains very many flexible regions, and to date has been tricky in order to crystallize in its entirety. Now, what we have done with the transglutaminase is, in fact, we were able to measure the conformational changes, both in the presence of GDP and in the presence of calcium. So in the absence of any small molecules, transglutaminase exists in 2 conformations, the open conformation and the closed conformation. We ran experiments on the sizing of transglutaminase and were able to get a sizing measurement of approximately 6.4 nanometers. And then what we did was we flowed in GDP as an analyte and measured the structure in the compacted form, and in this case sized at about 5.6 nanometers, and then we added calcium after washing away GDP and measured the expanded form at approximately 7.2 nanometers. And as you can see over here, we repeated the runs 3 times independently, and this is just to show what the spread of the data is, or what the deviation from one experiment to the next is. And as you can see over here, that there is very little deviation in terms of the sizing measurements that we were able to achieve. Now, of course, we didn't only run the sizing measurements in this case on the transglutaminase, but we also were able to determine the GDP binding kinetics. So we were able to measure both the association and the dissociation kinetics for this small molecule binder. So again, approximately 5 nanomolar KD, which is expected. Usually small molecules are not very tight binders to their targets. And for a lot of our sizing and conformational change work, we also have a couple of publications out there. And here I'd just like to illustrate one of these publications in which we ran the sizings of various different tRNA synthetase proteins. And based on the mutations of the tRNA synthetase, it was expected that they had a different hydrodynamic diameter. These measurements were conducted, again, in collaboration with people at the Scripps Research Institute. So for this and all further information and all further applications, I did not present them all today, but of course, if you're interested, please do reach out. All of this information can also be found on our website at dynamic/biosensors.com under our applications segment on our webpage. And with that, I come to the end of my presentation. I would like to start off by thanking the DBS Science Engine, all of our wonderful scientists that were responsible for generating the data that I was able to present to you today, and that are always excited to bring you the newest in applications and development. I'd like to give a special thanks as well to our DBS, our global sales team that is always, bringing all of our technology to the customers, getting the word out there to everybody. I'd like to thank our marketing team, especially my colleague Katerina, who did a lot of work in preparing all the materials that were brought to you today. And of course, a huge thank you to all of our audience. So thank you for joining us today. I certainly hope that you enjoyed this presentation. We really look forward to hearing from you. So please do reach out at any of these email addresses that we have listed up there. And let's see if SwitchSENSE is the solution for you. Thank you.